Okay, so we're going to talk about the last step in the central dogma today, and that is gene expression. Remember, the central dogma goes from DNA to RNA into a protein. So let's start our journey on gene expression. So, gene expression is a manifestation of a gene, which means that's the gene we actually see or what it's translated into. Most of the time, gene expression means that we're making proteins. Proteins are created through a process called transcription and translation, which involves an original DNA strand, RNA, and then it creates a protein from there. This process is really done in those two stages. Transcription is where we transcribe the DNA into RNA. It's like taking English and transcribing it into Spanish so somebody can understand you. So we're just transcribing it. It's the same words, it's the same verbiage, it's just in a different language. And then we translate it. So RNA will then translate that DNA code into what we know as a protein. So let's see what this looks like with the major roles of DNA and RNA. So there are so let's look at the difference between DNA and RNA first. So DNA is deoxyribose and RNA is ribose nucleic acid. Deoxyribose is the sugar within DNA, and ribose is the sugar within RNA. And we should know that OSE means it's a sugar. Both are still 5-carbon sugars, they just have a different bonding structure to them. Then we have our shape, a double helix for DNA, a singular helix for RNA. We'll also notice that we have thymine here, but in this one, we have uracil. Thymine is actually replaced to uracil over there. We still have adenine, guanine, and cytosine in the bottom. And so those 3 nitrogen bases stay the same throughout the entirety of it. However, we do see the uracil and thymine switch over. So our DNA, again, deoxyribose, the set nucleic, they still have those nucleotide polymers. Those are really important that we know. And a nucleotide is made up of phosphate group, a nitrogen base, and a sugar. Those are the 3 things in every single nucleotide. And again, adenine still pairs with thymine, guanine still pairs with cytosine in DNA, but in RNA, adenine and uracil pair together, while guanine and cytosine pair together. So there are 3 main types of RNA. The 3 main types of RNA are messenger RNA, transfer RNA, and ribosomal RNA, or mRNA, tRNA, and rRNA. The goal of messenger RNA is to transcribe the DNA sequence and carry that instructions into the cytoplasm, and where it'll be used for transcription, and then it's used during translation. The tRNA is just a transfer RNA. It's transferring amino acids. Remember, we have 20 amino acids in the human body that make up every single protein. It's transferring those amino acids in the correct order to create a polypeptide chain. And then the ribosomal RNA is really important. The ribosomal RNA, or the rRNA, actually binds to the messenger RNA in the cytoplasm, and it signals the transfer RNA to bring in the correct amino acid sequence. This way, your protein is made in the correct order. Without the ribosomal RNA, we wouldn't have protein synthesis occur. Oh wait, don't we have an organelle called ribosomes? Yes, we do. Protein synthesis now makes sense. So let's look at the structural components of this. The messenger RNA looks like a traditional RNA strand. It has a nucleotide sequence. We also see that it has 1, 2, 3, 1, 2, 3, 1, 2, 3. These are called codons. These codons are read by the rRNA, or ribosomal RNA here, and sent to the tRNA. The tRNA has anticodons, 1, 2, 3. 3 groupings of nucleotides create 1 codon. That will code for an amino acid. Every amino acid has its own specific sequence of codons. The ribosomal RNA, which is very globular, this will actually attach to the mRNA at the start site and create this process. So let's start about transcription. So this looks kind of familiar. Well, we still have a DNA strand and we have to access it. Unlike DNA replication, where a DNA helicase comes in and breaks the hydrogen bonds, and then a DNA polymerase comes in and reads it and reassembles, we just have one. It's called RNA polymerase. RNA polymerase is breaking those hydrogen bonds temporarily. It's resealing them almost immediately after, as you can see here. As it moves down the RNA strand or the DNA strand, it makes this new strand of complementary RNA. This mRNA strand is going to be the template that we use to create the protein. Now, we'll only do one side of the DNA strand and not the other side of the DNA strand. The one side that we're doing is specific for the gene that we're trying to create. And then we can have some mixed up steps in there if we do the wrong side. So here's step one. Translation begins with RNA polymerase binding to the specific sequence of DNA. We call that a promoter. We're promoting that site. That promoter site is the start site. From there, the rRNA polymerase will unwind the two segments of DNA, exposing it, breaking those hydrogen bonds temporarily between the two nitrogen bases. And then the RNA polymerase will add the complementary pairs to form the new RNA strand. However, as it reads, every time it sees an A, instead of putting a T as the complementary strand, it will put a U as the complementary strand. Transcription will follow the space pairs until we have a complete RNA strand. The RNA strand is only a segment of it. In DNA, we have segments called introns and exons. Some of them code for things and some of them don't code for things. So we need to remember when we're talking about gene expressions that we will have a coding segment and a non-coding segment. As scientists, we really don't know why certain segments are coding and certain segments are non-coding. They're kind of like placeholders. But for right now, we need to know that it's going to start at that promoter site and it will read until it hits a stop codon. That stops the process. And then we release that mRNA single strand and it will travel through the nuclear envelope and it will go into the cytoplasm. So transcription is a reverse of DNA replication. Remember, in DNA replication, we're using DNA as a template to create two new strands of DNA, where in transcription, we're not ruining that DNA strand. We're only copying a specific segment, and we have a new RNA template that's going to be removed and put into the cytoplasm. So I told you on the mRNA that they code for codons. 3 nucleotides together make a codon. This table right here is expressing that. So if we have the codon A, G, U, we want to look here. A is the first base, G is the second base, so we want to go over here, and U is our third base. We have the amino acid serine in this. You have a copy of this also within your textbook, located on page 307. You can use that as well. We do not ever use the anticodon. We're only using the mRNA strand, and it says it's the codons for mRNA. tRNA has the anticodons, and we can see serine is right here. If we look at this one, it's UAG, so let's go U, A, and G. UAG is what we call a stop codon. This is going to stop the sequence of DNA and not allow it to occur anymore. This is the end of the chain, allowing for it to release. Each of these 3 nucleotides is important because in the correct order, they make a known amino acid. But if they get rearranged, they can actually ruin the protein sequence that would occur. So the system of matching the codons to the nucleic acids is a really important one. It has to be done correctly. Otherwise, everything's lost in translation. Oh, wait, that's our next step, translation. Translation is basically converting it into a language that RNA makes the proteins. It's a different language, and it's not always interpreted the same way. just as you can say something in Spanish and then interpret it a little bit different in English. So we really need to be careful in the translation process so nothing is lost for the amino acids, because that can change the DNA or the protein sequence. So step 1, our tRNA kind of folds into a compact structure and attaches to an amino acid. The anticodon pairs with the amino acid. We have 20 amino acids in our body that make up every single protein. A lot of those amino acids have to be taken in through the food we eat or the nutrition that we take into our bodies. 8 of them can be made by our own bodies. The rest of them we have to get somewhere else. The second step is the tRNA molecule will bring it into the correct nucleotide. The rRNA is actually attached to the mRNA and is signaling out for the transfer RNA to bring in the right anticodon. The transfer RNA will attach temporarily and release itself. When it releases, it leaves the amino acid on the ribosomal RNA, and that's a really important thing to know. And it will repeat this until we hit a stop codon. Once it hits a stop codon, the polypeptide chain that's formed will release out, and the rRNA will also release. If your body signals that you need hundreds of these proteins, it will just go back to the start codon and start the process over again. There are 3 stop codons, UAG, UAA, UGA. Those are the 3 stop codons. If these stop codons are accidentally placed in the middle of a sequence, the protein that's needed won't be made. These newly formed polypeptides, again, will fall off the polypeptide complex, and they'll actually change shape, and we'll talk about that in a second. The last transfer RNA leaves the ribosome, and it will go back into the cytoplasm to find another amino acid. You got it. And at the end, if we need more, we just start this over again. So we can kind of see here what's happening. So we have our RNA polymerase breaking apart, making our new mRNA strand, which moves into the cytoplasm. The ribosomal RNA will bind to it, signaling for the tRNA to come in and bring in the amino acids, making a polypeptide chain, which we know as a protein strand. And so we'll watch it one more time. It moves into the cytoplasm. The tRNA is leaving the amino acids. They're leaving to go find some more, and they just create this wonderful little protein chain. That protein chain will then break off, and it will repeat translation as many times as we need to so that we have all of the proteins that we need. And you can have several proteins produced from the same RNA strand. Our bodies like to be efficient, so we can reuse until we don't need it anymore. The relationship between genes and the effects are really complex. Despite the neatness of this genetic code and the simplicity of this process and how orderly and functional it is, it's not quite the way it works inside of our bodies all the time. We have environmental factors that can kind of mess things up. An overall knowledge of the basic process of gene expression has allowed scientists to better understand how all working organisms work and why certain genes are expressed and why others aren't expressed. But the reality of it is we don't know why some people can make proteins all the time and some people can't. We're not really sure where all the proteins lie within the genome, which is a complete DNA sequence. And that's kind of crazy if we think about it. And it's something as simple as this. These simple amino acids, which we take in through our diet and our body makes a few of, we can go through transcription and then translation to create this little polypeptide chain. Poly meaning many. And then our bodies take that polypeptide chain and they twist it to form a protein that's a specific shape. That tertiary sequence of the protein is so extremely important because then it can be used for different functions within the body. And each one of these steps need to perform efficiently and effectively. Otherwise, you have a protein deficiency. And certain protein deficiencies can be life-threatening. In the next chapter, we'll talk more about gene expression and some of the reasons why our genes are the way they are and how a mutation can affect us. Until next time.